everyone back from lunch for our third session, and our first presenter is Nadia. Thank you.
Hello, I'm Nadia L'Bahy, a rising junior at the University of Massachusetts Amherst. And this summer I worked in the nanoquantum lab to determine if we could use bacterial cytological profiling to detect antibiotic-resistant bacteria. Since the conception of antibiotics, when Alexander Fleming developed penicillin in 1945, we've known of the potential threat of antibiotic resistance. Fleming himself even warned of the potential for bacteria to become resistant to our drugs in his Nobel lecture, when he described how people may easily underdose themselves and make their microbes resistant. Antibiotic resistance happens when you treat a population of bacteria with an antibiotic, killing off all of the bacteria except for those with a resistant or advantageous mutation. The red-resistant bacterium then has the chance to repopulate without much competition until all or most of the population is resistant. And it's really important that we understand antibiotic resistance because it's one of the largest medical concerns to date. So what can we do? We need a way of observing and quantifying the interaction between antibiotics and their bacterial targets, which is where bacterial cytological profiling comes in. Bacterial cytological profiling is a method that uses microscopic imaging to visualize the interactions between drugs and bacteria. It's cheaper and faster than sequencing, and we've already used it to develop models to determine the mechanism of action of different antibiotics. What is mechanism of action? Antibiotics can be grouped based on what cell components they target. For example, DNA synthesis inhibitors kill bacteria by impeding important DNA replication machinery, like DNA gyrase, whereas protein synthesis inhibitors target other cellular machinery and pathways. And it turns out that we can actually use BCP to accurately predict the mechanism of action of novel antimicrobials. When we image bacterial cells with BCP, we can easily see the phenotype of the cell wall in red and the DNA in green. This first image shows healthy wild-type strep pneuma, whereas the second image shows strep treated with levofloxacin, a DNA synthesis inhibitor. You can see really clearly how the normal wild-type pattern of DNA is disrupted in these cells. In cells treated with penicillin, a cell wall synthesis inhibitor, you can see really obvious differences in the shape and size of the cells. And you can also see obvious morphological differences in cells treated with rifampicin and kanamycin. These phenotypic differences are not only visible under the microscope, but are also quantifiable. We can process them into data that can then be analyzed using various models. So I spent the first half of my summer working on the model for predicting mechanism of action with BCP. And if you're interested in learning more about this, Loida has a poster on it in the atrium. So we thought if we can already use BCP to analyze drug-cell interactions, can we use it to detect when bacteria have become resistant to those drugs? To recap, we know we can use BCP to determine how cells are being impacted by antibiotics. The goal of my research was to then see if we could distinguish antibiotic-resistant bacteria from non-resistant bacteria, with the ultimate goal being to develop a model that could determine the frequency of resistant strains in a patient sample. Our first step was to develop adapted antibiotic-resistant strains of bacteria for experimentation. We treated wild-type bacteria with half of the minimum inhibitory concentration, or half of the concentration needed to halt growth. And we repeated this process until the population was completely resistant. We could tell that our populations had become resistant in part because of these growth curves. The graph on the left shows that before the bacteria adapted, their growth curve completely flatlines in comparison to the rapidly increasing no-drug control. Whereas after experimentation, once the cells have adapted, on the right, you can see that they grow just as well as the no-drug control when treated with 1 and even 2 times the minimum inhibitory concentration. Then we wanted to see if we could detect phenotypic differences between the adapted and non-adapted bacteria using BCP. When we look at the wild-type bacteria with BCP, we see these nice healthy chains and regular DNA staining. When we look at the wild-type treated bacteria, we see that they have been obviously affected by levofloxacin from the irregular appearance of their DNA. But when we look at the levofloxacin-resistant bacteria, there is no obvious phenotypic difference between the treated and untreated groups. So we can see that they're not being affected by the drug and that they have become resistant. And these phenotypic differences are not only easily visible, but they're also quantifiable. When we run a principal component analysis on the data generated by BCP, we can see how the different samples group together based on shared characteristics. In the levofloxacin sample, we can see the distinct brown cluster at the top of non-adapted treated bacteria, which is separate from the orange cluster of adapted, non-treated, and treated, which is also very far removed from the non-adapted, untreated cluster at the bottom. And we see these same clustering patterns in rifampicin, kanamycin, and penicillin-adapted samples. We then wanted to see if we could actually detect the presence of resistant bacteria in a population from this sort of analysis, and then whether we could accurately predict the percentage of resistant strains. So we carried out BCP on mixtures of adapted and non-adapted bacteria at known ratios. To do this, I started 2 different cultures of bacteria, one adapted and one non-adapted. I then split those into 2 groups each, one treated with antibiotics and one untreated. Then I mixed the adapted treated and non-adapted treated at known ratios, processed those cells, imaged them, and ran them through a profiling script that reduced their phenotypes to numerical data. Here's some of the data that I generated through this process. These are mixtures of rifampicin adapted and non-adapted bacteria at percentages of 0, 25, 50, and 100% adapted. After obtaining my images, I ran a script to process them into numerical data, or numerical representations of their phenotypes. I could then process that data and run it through a logistic regression that was trained to recognize 0% adapted and 100% adapted populations. That logistic regression could then predict whether each cell in a given image was adapted or non-adapted. These histograms represent the probability for each given prediction, where 0 represents adapted and 1 represents non-adapted, and the y-axis shows the frequency of each prediction. Everything in between 0 and 1 represents noise in our data. When we corrected for this noise, our predictions were a lot closer to the actual percentages. The leftmost column shows the percentage of resistant cells predicted by the logistic regression. The center column shows those values corrected for noise, and the expected rightmost column shows the actual percentage of resistance. So you can see that the difference between the predictions and actual ratios are pretty close to each other from this graph. We're also working on adapting this model and method for other resistant strains. This graph is a compilation of all the data I generated this summer. I ran it with 3 different antibiotics, levofloxacin, penicillin, and kanamycin. And for each experiment, I ran 8 different mixture ratios. I took at least 3 pictures per mixture ratio with around 1,000 cells per picture. So this chart represents a massive amount of data. And although you can see that the logistic regression predictions weren't very accurate, they do mirror the general upward trend of the actual percentages. So this gives us hope that by correcting for the noise in our data and maybe fine-tuning the model, we'll be able to generate more accurate predictions. So to conclude, we can use BCP to study the interactions between antibiotics and bacterial populations. We can distinguish between adapted and non-adapted strains of bacteria using BCP. But in order to detect the percentage of adapted bacteria in a given population, we need a model that sets a threshold to exclude noise and generate more accurate predictions. With that, I would like to thank Dr. Tim Van Opainen and the Boston College R&D program for having me this summer, as well as my 2 postdoctoral mentors, Dr. Alfonso Ortiz Marquez and Dr. Stephen Wood, my undergraduate lab partner, Loida Luna, and the NSF for funding me and giving me this amazing opportunity. Thank you. Questions?
I noticed that it seems like you think it's just the noise that's causing you to sort of, and is it even though you have the large degree in your expected versus your actual corrected number, that obviously the bottom end seems to be caused by your noise, but you're also not reaching that 100%. Why do you think you're not reaching that 100%?
So it depends based on the antibiotic that we're using, because obviously like rifampicin fared a lot better than the other ones, and that's because there are different mechanisms of action for all these antibiotics, and the different phenotypes can be more difficult to process with the model. So we don't know if we just need a different model that processes the data completely differently, or if it is just noise that's confusing it, but like I know with some different antibiotics, like kanamycin, it's a slower acting drug, so it may take longer time points to actually get like better data. Levofloxacin is really like this staining and coloration of the DNA, which is harder to quantify. So yeah, it might need to be on an antibiotic basis.
All right, our next presenter